Welcome to the seventh in this series of evolution lectures. I'm Mike Kasanovich, Director of Arizona Research Laboratories, and I'm standing in for our Dean of Science, Joaquin Ruiz. Tonight he's at another event, and it's a real pleasure for me to introduce our tonight's speaker, Dr. Michael Hammer. Mike is a small college guy from Lake Forest College, did his PhD at the University of California at Berkeley, went on to a postdoc at Princeton and another postdoc at Harvard, and unfortunately came to Arizona in 1991 to work in Arizona Research Laboratories as a research scientist. During his stay here, he's evolved his research to the point that he's now one of the world's leading experts on human origins, traced with DNA, and has made tremendous contributions. One way to look at this, he has appointments in Arizona Research Laboratories in Ecology and Evolutionary Biology, but he also, and I don't know why it's not on the slide, has an appointment in the Department of Anthropology. And that ties together his work and the impact of his work very nicely. I won't read you the title, you can read it, and ask Dr. Hammer to step up here and give his talk. Thank you.
Well, it's really a pleasure to be here and to have the opportunity to address our campus and our community on this topic of human origins using DNA to trace our origins. I've chosen to highlight three different themes in my talk tonight. The first one is our place in nature, and the second one is the evolution of the traits that make us human, trying to track both of those, not only genealogically, but in history as well. These are issues that Darwin addressed directly. And then the third theme will be something that's related, but something that Darwin didn't address, because this is something that has been debated most intensely in the last couple of decades. It's a topic that deals with the origin, how our species originated. The modern human species. And in order to do the work that I do and to address this issue in general, genetics isn't enough. It can't stand up on its own. We need different disciplines to work on this together to give us the full picture. We use paleontologists are involved in this topic as well. They study fossils to infer the morphological changes that have occurred over evolutionary time. Archaeologists who study material culture, stones, stone tool kits, to trace the history of our species, to infer the behavioral changes that have occurred through time. And then, of course, genetics, the area that I'm involved in, using both protein work and DNA work to infer our genealogical history. And one of the points I'm going to make in the talk tonight is that the genealogical history for different parts of our genome can tell us different things about our history. And that's one of the challenges that we face in trying to put together a story, a full story about our history. The history of the field of genetics actually goes back to, as applied to anthropology, goes back to the early 1900s in work that was done at the protein level and up through the 1960s. And I'll talk a little bit about some of the work that's been done with protein, and then the transition into DNA work in the 70s through the 90s, and studying individual genes and the sequences of those genes, and then in the last few years having entire genomes sequenced that we have at our disposal to make inferences about. Now I'm going to try and limit the technical information as much as possible, which is a challenge in itself, but I will probably convey a lot of the ideas to you through this drawing, these tree drawings. So on the left you can see a tree that may be a tree that was developed by anatomists, comparing the anatomy of humans and great apes and inferring their relationships. That's a species tree. In the middle I'm trying to convey to you this timeline of fossils as they've been discovered and dated, creating paleontologists will infer the taxonomic status of a particular fossil by comparing it with other fossils, its time depth in the fossil record, and make inferences that almost look like a tree. You can see these branches that I've shadowed underneath these timelines of fossils. And then on the right is a tree that is a gene tree. It's not a species tree, but it's a tree that reflects the genealogical history of a particular region of the genome. And the time frame for the genealogical history can come from the number of mutations that we see in the DNA, using some kind of a molecular clock approach to infer the time depth of the tree. Now, let's turn to what Darwin said about human evolution. We can summarize it in one sentence. In his Origin of Species from 1859, light will be thrown on the origin of man and his history. That's all he said in the Origin of Species. And if we look a little further in the literature, we find that he wrote to his friend Alfred Russel Wallace a couple of years before he published the Origin of Species, in 1957, when asked would he address this issue of human evolution. And what he said was, "I think I shall avoid the whole subject, as so surrounded with prejudices, though I fully admit that it is the highest and most interesting problem for the naturalist." But his ally and bulldog, Thomas H. Huxley, just 4 years after the publication of The Origin of Species, published his own book called Evidence as to Man's Place in Nature. And he wasn't shy about tackling this issue about human origins and applying evolutionary theory to human origins. What he did was he did an extensive comparative analysis of human and ape anatomy, embryology, and ethology, behavior, as well as looked at the very, very few fossils that were known at the time, and concluded that humans have a close evolutionary relationship with the great apes, in particular the African apes. So this was striking for many reasons. At the time, there were many out there who didn't believe that humans had any relationship at all to the rest of the biological world, number one, but then to claim that humans were related to African apes as opposed to the orangutan, Asian ape, was quite a striking statement, a bold statement. But perhaps the biggest impact would be in a philosophical sense. Humans now were seen as part of nature and not apart from nature. In fact, the Darwinian revolution followed by a couple of centuries another revolution in Western philosophy, stirred up by Copernicus, who gave us the heliocentric universe, disposing of the geocentric universe where everything circled around the Earth. Earth now circled around the sun and was just one planet of many in the universe. It was no longer the center of the universe. And similarly, Darwinian revolution put man from nature, took man from a position out of nature into a position in nature, made the naturalistic view of man. But still, the human in either case still remained at the pinnacle of God's works. And so these revolutions are in the context of these other kinds of thought processes of natural philosophers at the time. But still, Huxley made a statement that gives us a picture of the kind of tension that existed between the continuity of placing man in nature and the discontinuity of man from the rest of nature. In this quote, "No one is more strongly convinced than I am of the vastness of the gulf between man and the brutes. For he alone possesses the marvelous endowment of intelligible and rational speech, and stands raised upon it as on a mountaintop, far above the level of his humble fellows, and transfigured from his grosser nature by reflecting, here and there, a ray from the infinite source of truth." So what I'd like to talk about tonight is a bit about the tension that's shown in this statement. What is the relationship between man and the rest of the biological reality? And follow the history of that and see what DNA and molecular approaches have done to help us understand that relationship. And in the first two themes that I'm going to be developing, the first is, what is the relationship of man to the great apes? And I use the word man in the 18th and 19th century sense. Another way of putting that question is, how far back in time did humans and great apes share a common ancestor? That's theme number one. Theme number two is also something that has been debated greatly in the last century, and that is the question of humanness. When did the traits that make us so special and so different from the animal world, when did those evolve? And when we answer that kind of question, we think about all the changes that occurred on the lineage leading specifically to modern humans. This lineage here, and I've shown some fossils that represent extinct forms of humans or pre-humans on the lineage leading to modern forms. And I will refer to all of the creatures on this branch as hominins. Now, these two themes in practice are deeply intertwined because in order to determine what makes us special, we need to know who to compare ourselves with. So, what is the history of this idea of establishing the link between humans and the great apes? As I just mentioned, Darwin and Huxley both, through their analysis, reasoned that humans were closely related to the African apes. Whereas the view seemed to shift. After 1920 or so, up until 1960, the view shifted back to one where humans were much more distantly related to the great apes. In fact, it was seen that the great apes, the African apes, and the orangutan were closely related, and either of those being more related to each other than any to humans, humans being very distinct. And one version of that, this is work that was written in the 1920s as representative of the kind of things that were being said in those days, work by Sir Arthur Keith. He showed this family tree here of the great ape lineage here, the human lineage, and seeing how far back in time they coalesce back to a common ancestor in the Oligocene, which today we know to be about 30 million years back in time. Along with this view of the ancient split between humans and the great apes was the view that human populations themselves traced back very far in time, so that the different racial groups were very old. And these were very consistent with the racial views of the time, that the white race had reached ascendancy more recently than these other races around the world. This was a common view at the time. So perhaps the most important fossil support for this ancient divergent hypothesis was not that old in time. It was a fossil called Ramapithecus that had been discovered in the 1920s and rediscovered by Elwyn Simons. Ramapithecus is an ape-like creature that lived in Eurasia about 15 million years ago and appeared to be anatomically related to the hominid lineage, according to Simons. And based on these similarities, he suggested that this lineage had the Ramapithecus on its right here on the lineage, which would support this very ancient divergent between humans and the great apes. Well, let's now look at what the molecular field has done for us. In 1962, the term molecular anthropology was coined by Emil Zuckerkandl and Linus Pauling, who were the first to suggest that one could use molecular approaches to infer evolutionary history. By comparing the sequences of proteins extracted from different organisms, one could then infer the evolutionary relationships of those organisms. This launched a new field called molecular anthropology, which was really christened by Morris Goodman in 1963, and then Sarich and Wilson in 1967, who made immunological comparisons among the proteins of African apes, orangutans, and humans, and showed that, yes, humans were more like African apes than they either was to orangutans. Goodman's analysis was qualitative. Sarich and Wilson's analysis was quantitative. And this shows you the result. Darwin was right, Huxley was right. According to the molecular approach, humans share a relationship with African apes that only goes back 5 million years. And this was the Sarich and Wilson result, the quantitative result, suggesting an incredibly recent divergence between humans and the great apes. In fact, this conclusion was heretical for a number of years and wasn't fully accepted by the anthropological community until additional fossils had been found, which gave us a better view of where Ramapithecus fit onto the tree of the hominoids. In fact, Ramapithecus now is classified as a fossil ancestor of the orangutan. And so all of the picture makes sense. And in this case, the molecular studies led the way in seeing the right answer, if we ignore what had been said by Huxley and Darwin 100 and some years before that. But notice on this tree on the right that we have basically a three-way split, or a trichotomy. How can we resolve this trichotomy? There are a number of possible ways it could be resolved. In terms of, it could be a true trichotomy. It could be that these three species actually diverged so close in time that essentially they are all equally related to one another. Or you could have any of these other permutations where gorillas and chimps are more closely related to each other, or one of them related to humans more so. In order to resolve this, a great deal of genetic information was necessary, because it is actually a very small period of time in which these three species separated from one another. And in order to solve this problem, we required the new technology of DNA. And there were two early pioneers in this regard, Sibley and Ahlquist, who explored DNA kinetics and how by hybridizing the genomes of different species one could determine their relationships. And then there was the work by Morris Goodman by looking directly at the DNA sequence level, looking at enough genes, enough sequence lined up from chimps, gorillas, and humans, one could infer who was more closely related to the other. In fact, when this work was applied, the answer came out in both cases, for Goodman and for Sibley and Ahlquist, to show that humans were more closely related to chimpanzees than either was to gorilla. In fact, these studies were right on the mark in showing that humans and chimps shared about 98.8% of their DNA sequences. So that's fairly striking. So if we kind of now look back at the history of the idea of man's place in nature, we can see that it's really characterized by a series of demotions, starting with humans having a special creation, to humans having a special branch on the tree of primates, to humans being one of many branches on the tree of primates, to humans and chimps being part of the same genus almost. So this is striking information and had striking consequences for how we view ourselves and our place in nature. In fact, because now we can look in the genome to see what makes us special, we have a special trick we can use. We can look to the chimp genome to see where the differences are and what differences may have arisen that do make us special. Now if we move to theme number 2, trying to track how far back in time these special traits may have evolved that make us human. Darwin had a view about this as well. Basically, Darwin figured the first steps on the way to the human lineage, when apes first budded off into this group that eventually became humans, that brains led the way. He reasoned that a smarter ape was the thing that would eventually get us to being humans. The other view was that walking led the way, that standing upright would be the first step in the early process of becoming human. In fact, Darwin had a good reason to put forward his idea. He reasoned that in the descent of man, those characters that make us human, he identified as intelligence, manual dexterity, technology, and uprightness. And he argued that an ape endowed with minor amounts of intelligence or any of these other characteristics would surely give that ape an advantage over other apes. And a forebear endowed with these qualities then would just inevitably become human through the power of natural selection. In other words, Darwin thought that hominin origins equaled human origins. For those of you who don't recognize, this is the monolith in 2001. Well, Darwin didn't have fossils at his disposal to test his hypothesis. So it wasn't until many years later, in the 1940s, 50s, and 60s, when we had a huge number of fossil discoveries that allowed us to piece together the history of the origin of these different forms that eventually became modern humans. And so this slide shows you a summary of the current view from the fossil evidence with some of the key players on the sides. You have Homo erectus, Homo ergaster, and Homo habilis shown here. These are all part of the genus Homo, which is shown in blue. And then you have Paranthropus boisei, robustus, and Ethiopicus, which are shown over here. They're later bipedal apes. And the reason I contrast them is because you can see they lived actually at the same time. Darwin also had the idea that humans evolved along a single lineage. Well, with this number of new fossil discoveries, it became apparent that there were multiple species that coexisted at the same time. And you can see that boisei here with the large sagittal ridge on his head, and you can see over here erectus, could not be characterized as the same species. But more importantly, what we can see from this ordering of characteristics is that the earliest hominids leading up to the Australopithecines, Lucy is the most famous of the Australopithecines, essentially were small-brained creatures with large teeth, and clearly proved Darwin to be wrong. These earliest members of the hominid lineage, in fact, could be seen as bipedal apes, with the quotation mark around apes, since we're talking about the hominid lineage. We're no longer apes, but they looked a lot like apes, but they were able to walk. These bipedal apes, these early bipedal apes, eventually gave rise to more robust forms, called Paranthropus, and more gracile forms, the genus Homo. Eventually, the genus Homo led to Homo sapiens here at the top. And you can see how recent in time our species, Homo sapiens, emerges in the scheme of hominid evolution. So we can say, by looking at the fossil record, that we no longer would consider Darwin to be right in this count. Hominid origins are not equivalent to human origins, and we must think now of the early evolution of our specialness being one of being upright, where the larger brains came later. Most of us, I think, would say that part of what makes us special are the cognitive abilities that must relate somehow to our larger brains. But we're not quite there yet, because this slide is showing you sort of the encephalization quotient, which is a way of giving you the relative brain size to body weight on the y-axis and the time scale on the x-axis. You can see that if we compare ourselves to chimps, we have a larger brain relative to body weight at the top, and then the various fossil species that I talked about earlier in the middle. And you can see that the so-called bipedal apes have a slight increased brain size over that in chimps. The intelligent omnivores, those early members of the genus Homo, had a slightly larger brain size again, but neither reaches the brain sizes that we have at the top. So in order to get to the characteristics that really make us human, we have to go on to the next stage of evolution and talk about the transition to anatomically modern forms. Also, the toolkits of the various species that I'm talking about. It turns out that all of the species I've discussed have been using basically the same set of tools. In about 2.5 million years ago, the first stone tools are found in the archaeological record, known as Oldowan tools, which eventually, about 1.7 million years ago, become a slightly more complex grade of tools called Acheulean tools. Here you have Acheulean toolkits, and you can see basically what they look like. It's hard to tell a hammer from a crescent wrench. So we're not there yet archaeologically, which is a reflection of where we are cognitively at some level. So in order to get into that and understand how we made the final transition to the Homo sapiens, the species that we are, I would like to bring up theme 3 of my talk tonight. How did we make the transition to modern form? Homo sapiens shown up again on the upper left, and now I've got some different players here, all in the genus Homo, and you can see Homo sapiens, or anatomically modern humans, shown in the upper left, has a much larger cranial capacity. It's pretty obvious when you compare the relative sizes of the cranium versus the upper and lower part of the cranium. I also want to point out again that there are multiple species or taxonomic groups living before Homo sapiens, and even after Homo sapiens emerges, there are others that coexist with Homo sapiens. Homo neanderthalensis, which lived in Europe, Homo heidelbergensis, which also lived in Africa and Europe, Homo erectus, which lived in Asia. Homo erectus is thought to have originated in Africa from a precursor that may have been very similar. There's debate as to whether there's Homo erectus in Africa or Homo ergaster. I'm going to go with the terminology of Homo ergaster, giving rise to Homo erectus in Asia. And then this new find here, Homo floresiensis, which I'll point out in a minute. Now this slide is supposed to represent the very recent origin of Homo sapiens, showing you in the geographical context. Notice after Homo sapiens originated, Homo sapiens rapidly colonized the globe in the last 100,000 years. But Homo sapiens was not the first species to migrate out of Africa. You can see that Homo erectus, shortly after it emerged in Africa, migrated out and colonized parts of Southeast Asia and East Asia. Now this map shows you the distribution of some of these fossil finds for Homo erectus in green and the Neanderthal range in blue. And what I wanted to point out is you have some of the earliest Homo erectus or ergaster fossils in Africa about 1.8 million years ago. And then out here in China, 1.2, in Dmanisi, Georgia, 1.7 million years ago, and here in Java, 1.7 million years ago. So shortly after the first Homo erectus is known to exist, we find fossils widely dispersed across the globe, at least the old world. And I also wanted to point out that Neanderthals may have been around until about 28,000 years ago, certainly coexisting with modern humans in Europe. And in Asia, we have erectus existing on Java till 25,000 years ago, again coexisting with moderns. And then this find of the island of Flores in Indonesia is quite interesting. These are people who were no more than 3 feet tall, sometimes called hobbits, that were probably related to Homo erectus, descended from Homo erectus, and then underwent what we call island dwarfism. And they look like they have lived up until as recently as 13,000 years ago. So one of the questions that's so staring us in the face is, we are alone as a species today, and yet just as recently as 10, 20, 30,000 years ago, we shared the planet with other forms of humans. Why did this happen? And why are we alone today? That's an open question and a mystery that I will try and address a little bit through the rest of the talk. Now I want to talk about a couple of different models for the formation of our species. There are 2 models that have been hotly debated in the literature for the last 20 years, both of them based strictly on fossil evidence. The one here on the left that is called multi-regional evolution suggests that when Homo erectus dispersed out of Africa, colonized Asia, from that population, which remained fairly continuous across that geographic space, gave rise to Homo sapiens pretty much everywhere that it occurred. In this model, there's no speciation. There's some local differentiation among Homo erectus populations, but when a novelty would arise, such as a gene that might encode a larger brain, this gene would then be free to migrate from one population to the other, and perhaps with the advantage of natural selection, spread throughout the range of Homo erectus. So that Homo erectus made this transition to the modern form in concert across the globe. This idea has its roots going back to Darwin, in a sense, who believed in gradual evolution of a single lineage. This model sees Homo as one species. It makes the predictions, well, I'll talk about the predictions in a minute. The other model is the recent African origin model, which says that modern humans originated in a single place in the world, very recently, Africa. And from that African place, came out of Africa, replacing all the other non-modern forms that existed in Europe and in Asia. This model suggests that all the genes that make us modern evolved in one geographic location, somewhere in Africa. The model also specifies the population was relatively small and isolated. And this model has its roots in a different philosophy in evolutionary genetics, that of one where you have lots of speciation. The boundaries between populations are hardening up, and so to speak. Genes are not as permeable across populations because they're beginning to speciate. It's a model of cladogenesis. Now, what are the predictions these different models might make that we could actually test using genetic data? Well, the multi-regional model, as I said, makes two predictions, one about place, one about time. I'm going to try and keep it as simple as I can. In this model, you can see multi-regional evolution. These lineages all descend back to Homo erectus time, and the transition to modern is shown by this change from red lineages to yellow lineages. But if you look at the lineages today, we should be able to trace them back very far in the past, and they should be able to trace deep in Europe, deep in Africa, and deep in Asia. So we might say the lineage will trace to many parts of the world. That's the place. The time, they could be going back, many of them, over 2 million years. For the recent African replacement model, we have an African population that contains all the genetic variation of subsequent descendant populations. So all of the lineages that we're going to trace that are yellow, the modern lineages, are going to really trace back to one place, Africa, and they're going to trace back to Africa much more recently than the lineages would trace back to a common ancestor in the model, in the multi-regional evolution model. So we'd say the lineage traced to Africa in the more recent past. So now I want to use these predictions, time and place, the genealogical depth of different parts of the genome, and ask, do the lineages of different parts of the genome trace to one place, and how far back in time do they trace? To see if we can answer this question about how we emerged as our species. Now, in order to do so, I just need to tell you a couple of things about the genome that we're going to be looking at. There's essentially two genomes. In every cell, in almost every cell, blood cells don't have a nucleus, but most cells have a nucleus and a cytoplasm. In the cytoplasm of a cell, there's an organelle called the mitochondria, which has to do with producing the energy for the cell. And it contains its own small circular genome. It's only 16,569 bases long. It's just over 16 kb in length. And it's inherited strictly from mother to children. And the nucleus is the rest of our genome. It's 3 billion base pairs. There's 23 different pairs of chromosomes. The first 22 are called autosomes. We get one autosome from mom, one from dad. And then the 23rd pair is called sex chromosomes. You have an X chromosome and a Y chromosome if you're a male, and you have 2 X chromosomes if you're a female. Now, this slide's a bit tricky to see, but I've got 4 generations, great-grandparents, grandparents, father and mother, and a son at the bottom. And what I've shown here is his mitochondrial DNA, this small circular molecule here, his Y chromosome. And you can see that his Y chromosome traces back to one male each generation, to his father, to this grandfather, and to this great-grandfather. Whereas mitochondrial DNA traces back to this mother, to this grandmother, and to this particular great-grandmother. So none of the other great-grandparents in this generation have contributed their Y chromosome or their mitochondrial DNA to this boy here. However, if you look at an autosomal pair, I've just chosen a single pair for the sake of simplicity, you can see that this patchwork of different colors and patterns represents pieces of autosomes that came from different ancestors in the past. So you can see that this piece of his autosome traces back to this grandmother, and this piece to this grandfather, and so on. So what I'm trying to say here is, because of the process of recombination, pieces of the genome are shuffled between chromosomes that are inherited from father and mother. Each piece of the autosome may trace back to a different ancestor, whereas the Y traces back to one, the mitochondrial traces back to one female ancestor in the past, all the way back to a single common female ancestor that is the lucky mother of all of our mitochondrial DNA, or the lucky father of all of our Y chromosomes. The same is not true for the autosomes. And that's a point that I can't emphasize enough, and I will try and bring up again to tell you what this means for our evolutionary past. So for technical reasons, the first part of our genome that was studied and addressed the question of human evolution was the mitochondrial DNA. Because it was isolated, it was small, it was in the cytoplasm, we could isolate it experimentally and study it back in the 1980s. And we were able to get DNA sequence information. There was a landmark study by Rebecca Kahn and Alan Wilson at Berkeley in 1987 where they surveyed mitochondrial DNA variation in many people from Africa, Asia, and Europe. And what they inferred was that all the mitochondrial DNA in these people they looked at today traced back to a single woman who lived in Africa. And I think it was the press and not the scientists who dubbed that person Mitochondrial Eve. It gives us that strong metaphor of tracing back to a single woman in the Garden of Eden, which is a rather unfortunate metaphor in some regards, because the work did not imply that there was only a single woman living at the time, but there was just one woman who was the ancestor of all mitochondrial DNAs today. This work was reproduced, in a sense, in the year 2000 by Ulf Jelinsson and Max Ingman in Sweden and colleagues. They looked at the entire mitochondrial DNA genome. They sequenced the entire genome in 53 different individuals, also from Africa, Asia, and Europe. And I'm showing the tree that they were able to infer from the data. And what you can see here is that you have African lineages on the bottom and Eurasian lineages on the top. And you can see that the African lineages on the bottom are longer, deeper. And so this reflects part of the inference that they made that the ancestor was in Africa. They found greater African diversity. They found that the place of the ancestor was Africa, and that they found the time of the ancestor was recent, only going back about 170,000 years. So those are the predictions of which model? The recent African replacement model. Another very intriguing piece of evidence comes from Svante Pääbo and his collaborators, their work on ancient DNA. They were able to isolate a small amount of mitochondrial DNA from Neandertal fossils. And as you can imagine, this is a tricky enterprise. Not only does the DNA degrade over time, but all the people who've handled the bones or all the DNA that's floating around in the laboratory is just as easily studied as this tiny, tiny amount of Neandertal DNA, unless you're very careful not to contaminate it. But they were able to show conclusively that the mitochondrial DNA from their extractions of this Neanderthal bone were, in fact, Neanderthal DNA. They looked at this, they did the experiment multiple times for multiple Neanderthal individuals, and they showed on this tree over here that the Neanderthals were all very closely related to each other, and yet very different from the huge number of sequences that had been examined in humans at that point, suggesting that there was no Neanderthal mitochondrial contribution to the human gene pool. Again, this is a prediction of the recent African replacement model, whereby African ancestors make the transition to a modern form and then spread out, replacing all the archaic forms. This is what I'm calling the mitochondrial paradigm, because this work was so powerful. It came at a time before we had studied much of the rest of the genome. It came at a time when archaeologists and the paleontologists were making these debates based on fossil evidence, and they weren't getting there anywhere with their arguments. And this seemed to be the first genetic evidence that came down very strongly on the side of the recent African replacement model. This is an image from the cover of Newsweek in 1988. They're flirting with this idea again of African Eve, mitochondrial Eve, and the search for Y chromosome Adam. And this diagram on the left shows you, also taken from Nature magazine, a summary of the results that were published by Kahn et al. in 1987. Now, it was clear to many of us working in the field of genetics that mitochondrial DNA is just one little tiny part of the genome, and that we really need to investigate many other parts of the genome to see if we would find similar patterns. The most obvious next place to look was the Y chromosome. And this reflects work that my lab has done and another lab at Stanford over the last decade or so, where we were pioneers in looking for variation on the Y chromosome and tracing variation to creating these genealogical trees to see what it told us about human evolution. And what I'm going to say is, it's very simple. The pattern is almost identical to what had been found from mitochondrial DNA. Number one, you can see the tree has the African lineages that are longer than the non-African lineages. You can see that African diversity is greater. The place of origin is Africa. And the time to the most recent common ancestor for the Y is even more recent than for the mitochondrial DNA, only going back roughly about 100,000 years. Remember, the mitochondrial DNA traced back about 170,000 years. So both systems are telling us pretty much the same thing, But the fact that they trace back to different times is quite interesting. It just highlights that each part of the genome traces back to a different ancestor, and that ancestor may have lived at a different time or a different place. In this case, the place happens to be the same, Africa. And what we want to know is, if we looked at many parts of the genome, would they all trace back to ancestors that lived in Africa, consistent with this recent African replacement model? Well, that work was done in the middle 90s and the late 90s, and by this time, new technology had emerged that allowed us to examine greater portions of the genome more efficiently, and we started to build up a lot of data on the autosomes and the X chromosome. So the rest of the talk, next section of the talk will be telling you something about this huge body of work. So I'm going to try and summarize it very briefly. First of all, if we look at genetic diversity on the X and the autosomes, compared that level of diversity between Africans and non-Africans, you can see something that's familiar to you. There's greater diversity on African populations than in non-African populations for both autosomal and X chromosomal data. There's about a 30 or 40% reduction in non-African diversity, perhaps hinting again at this older population in Africa. Maybe our autosomal and X diversity is reinforcing this idea of an African origin. Then there was a fellow named Yuki Takahata, a Japanese fellow, who wanted to do his own analysis of the nuclear genome, which is much more difficult to do because of the recombination obscuring the history of these lineages over time. But what he was able to do was find a number of studies of the autosomes and the X chromosome, independent regions, different gene regions that are shown in the column on the left. The top two columns are the Y and the mitochondria. It's just review for you to see that they also trace to Africa. But many of the regions on the X and the autosomes also trace to Africa, but notice that not all of them do. Here's a region that seems to trace back to an African ancestor. Here's another region that traces back to an Asian ancestor. Here's one that traces to a European ancestor. I'm sorry, there are two tracing back to Asia, and the rest are tracing back to to Africa, and there's one tracing to Europe or Africa. So the general pattern is, yes, a lot of our variation in the nuclear genome is tracing to Africa, but there are exceptions, and I don't want those exceptions to get lost. I will talk about them explicitly in a minute. But let me talk a little bit now about some of the differences between the mitochondrial and the Y chromosome results and the autosome and the X chromosome results. I've shown, again, this fossil timeline of our ancestry on the right, and on this side I'm showing you the time at which these different parts of the genome trace to an ancestor. So I've already mentioned that the Y and the mitochondria trace back to an ancestor very recently, within the last 200,000 years, and I'm showing that here. Notice, for the X and for the autosomes, the mean time for many different studies traces these parts of the genome back almost 1 million years or more. Sorry, just lost the slide for a second. So this is the average time back to an ancestor on the X and the autosome, almost 1 million years. And the vertical bar, shows some of the range of estimates back to the ancestor, some of them going back to almost 3 million years ago on the autosomes. So this is time going back to time when Australopithecines were still walking around in Africa. But even the means are telling us something much deeper in time than these haploid or Y chromosome and mitochondrial regions are telling us. Just to highlight what some of these patterns look like, this is work that's been done in my lab in the last few years, where we looked at a bunch of different regions on the X chromosome. I'm showing the X chromosome lying on its side down here on the X-axis, 16 different regions of the X chromosome. And I want to emphasize that these regions are unrelated to each other. They are independently evolving regions because of the process of recombination. And so when we looked and estimated the time of the ancestor for these 16 independent regions and plotted them out along the chromosome, you could see they varied quite a bit from pieces of the DNA that trace back to an ancestor 400,000 years ago to some that trace back as far back as 2 million years ago. There's no particular pattern when we look at them as spread out on the X chromosome as they are, but you see this variability. If we plot those times against, now not plotting them against the X chromosome, but plotting them against just an axis of time going from 200,000 years back to 3 million years, you can see that the observed values that I just showed you on the previous slide give you a bimodal distribution of times back to the ancestor. this group here and this outlier group here. And these are the observed times to the common ancestor that I just described. This curve here represents the expected time back to the common ancestor under a recent African replacement model. So if we were able to simulate a recent African replacement model and ask what is the, and you do this for many different regions of the X chromosome, we'd expect most of the regions on the X chromosome to trace back to an ancestor about 600,000 years ago. But then there's this tail where we might expect some to trace back further in time under the same model. But if you notice here, this second little group in this bimodal distribution are on the curve of this expected, on the tail of this expected curve. They're outliers, and they're not fitting the model exactly. So if we were to summarize what this data were telling us, would be that most of the data are consistent with this expected curve, the model of the recent African replacement, but the recent African replacement model can't explain all of the data. There are some outliers. And so we wanted to know what these outliers were, what was going on with them. And so one of them, I'm going to tell you one of the stories of one of these outliers. This is a region on the X chromosome way out near the tip. It's a region of DNA that has no particular function right now. It's a pseudogene. It once functioned as a gene, but it experienced some knockout mutations. It no longer functions. So it's evolving in a very neutral way. We don't expect it to be affected by natural selection. And Dan Garrigan in my lab was heading up this study. And here's Dan. I couldn't find another picture of him other than the one riding his dog. But the pattern we saw on this particular region of the genome was completely different than what I was showing you for the mitochondrial DNA in the Y chromosome. If you noticed, before there were Africans on one side of the root of the tree and everybody else on the right. Here you see Asians on one side and Asians and everybody on the other side. There was greater diversity in Asia as opposed to Africa. The place of origin is Asia, and the time of the ancestor is almost 2 million years ago. So we wanted to know what this meant. Here's a way of portraying that genealogy for that gene right here on the right against the fossil record again. And you can see that time of the ancestor back here at the root of this tree corresponds very nicely to the time when Asian erectus and African ergaster were splitting, diverging. And so we think that part of the genealogy of this particular locus in the genome is reflecting erectus ancestry. So how would we have some erectus ancestry in our genome? Well, this is a slide I've taken from a paper first reporting the Floresiensis finds. I thought it was very fitting because what they did was they showed how the finger leading from Homo erectus to Floresiensis, bringing it right up, overlapping in time with Homo sapiens. If we just imagine that as a potential for interbreeding, then we could imagine a scenario where as modern humans came out of Africa and colonized Asia, Southeast Asia, found themselves face to face with these archaic forms, there may have been some interbreeding. And this, depending on what level this occurred, could have allowed certain of erectus genes to filter into the human gene pool. So this was probably the first evidence we had of something like this. There's more evidence accruing as we speak. There are other studies shown in the literature where we have these ancient diverged lineages that don't fit the recent African replacement model. They certainly don't outnumber the number of loci that do fit the model, but there is certainly something we need to pay attention to in understanding what's going on. So because we have these different patterns, and each region of the genome is telling us a slightly different story, I like to think of the genome as a mosaic. It's an evolutionary mosaic. That if you look at one region of the chromosome, it'll have a certain genealogical pattern. Maybe it's tracing deep in time to a certain part of the world. If you look at another region, it may have a completely different pattern. It may be tracing more recently to a different part of the world. Or it may be that that particular region has undergone adaptive change. It's reflecting the process of natural selection. But in any case, we can't think of the genome strictly in terms of tracing back to a single ancestor. We have to think of it in this more complex way. It makes our analyses more difficult, but I think it's shedding some light on some very complex processes that were going on at the time that Homo sapiens emerged. So how would I go back and say, what's the best model to explain what we've observed with the data? I would say that neither of the extreme models I presented can explain all of the data, but if we come up with some kind of intermediate model, borrowing aspects of those two extreme models, we might very well be able to explain the patterns we see. So if we borrow the part of the recent African replacement model where populations are isolated and tending to speciate, tending to be different enough so they no longer interbreed, but able to exchange some genetic material, gene flow between populations, which we borrow from the multi-regional model, we could explain perhaps the bulk of our genome coming from one place, Africa, more recently, with some leakage of these more divergent lineages penetrating into our genome. And so that is the current view that I think explains all of the data best. So what it means is that for the bulk of our genome tracing to Africa, recently, we're mostly modern Africans, wherever you live, whether you live in Africa now or whether you've been in exile from Africa for a while. We are essentially modern Africans, all of us. Except there is some part of our genome that may reflect something else. And I don't know exactly what that is, but if it's a problem for you, there's a solution. Maybe that's not so surprising to some of the women in the room who wondered why their husbands were the way they were. And just to summarize, we started out asking, how far back in time did our special features as human evolve on the lineage? It's clear that we've experienced a stepwise evolution of our specialness. We started out as bipedal apes. We eventually got bigger brains, and we eventually became modern, got bigger brains yet, and acquired the cognitive abilities that we have. We don't see the appearance of things like cave art until 35, 40,000 years ago, but we do see in Africa the very early underpinnings of art, of culture, in the archaeological record. And this is not a very pretty slide. I just grabbed it off the internet about 2 hours ago. I didn't have a chance to make it look pretty, but we're tracing some of the earliest, through the archaeological record, things that you might qualify as art. Painted beads and things like that, that trace back in Africa further than they trace back anywhere else on any other continent. So just like the genome, the bulk of our genome tracing to Africa, it seems that the majority of our cultural heritage also traces back to Africa, but only going back on the order of 100 or 200,000 years. So the qualities that make us special are very new in evolution. And I think we can meditate on that, and I'll thank you. Take questions.
Thank you, Dr. Hammer. In a book I recently read called The Journey of Man, the author uses what he called DNA markers to trace the geographical migration of man. Can you explain to me, please, what a marker is?
I'm sorry. Can we? I'm losing your question.
Journey of man, using DNA markers to trace the journey of man. Can you explain that?
Can I explain that? You know, the journey of man and the markers that are used to study that essentially are the Y chromosome and the mitochondrial DNA, those two markers that I told you about first that trace back about 170,000 years ago or 100,000 years ago and reflect very nicely the colonization of the world that occurred after modern Homo sapiens emerged about 200,000 years ago. So that part of our journey is something only going back 100,000 years or so, and the mitochondrial DNA and the Y chromosome are very well suited for tracing that journey. But for tracing the journey that was before that, preceded that, this entire history that lies below that, we must look to the nuclear genome, because that's the only place we're going to have the genealogical depth to make inferences about ancient history.
Doctor, Okay. I read a review recently of a book called Demonic Males by a professor at Berkeley, and he was highly dismissive of the idea that we share 98% of our genes with chimpanzees. Was he just being a crank or what?
Demonic males? Was that the title of the book?
I highly recommend the book.
I'm sorry. So the question was that there's a book written called Demonic Males?
Yes, but I'm
concerned. The author questioned the relationship with chimpanzees?
Yes. And he's a professor at Berkeley.
Well,
he says that Sibley and Alquist got it wrong.
well, I think that there's too much evidence now from the whole genome. We've now sequenced the entire chimpanzee genome, the entire human genome. So if you had any problems with Sibley and Alquist's experiments in 1984 or whatever, I think that everything that's happened since then has verified their results. And I personally can speak of aligning sequences from humans and chimps and counting the differences myself. So the data from my lab don't lie. I don't know about anybody else's.
Back on the slide that showed, I had the map and where each of the fossils were from, and it said that over in Indonesia there were the species that might have existed as recently as 13,000 years ago. It showed in green and purple the different locations of the fossils, and there was a little bit of green up on the bottom of the British Isles. What is that representative of?
Yeah. Well, I don't know how what's that green doing in the British Isles? There is some speculation that Homo erectus got back into Africa and up into Europe and made it up as far as the British Isles. But there are also other fossils that I haven't put on here. Boxgrove, Homo antecessor, Homo heidelbergensis, that found their way into Spain and Western Europe. And they were transitional. They weren't modern. So I think maybe that little patch of green might be the Brits' version of archaic. We shouldn't debate how far that green goes up the British Isles, though.
I have a couple of questions about taxonomy. When you say we're modern Africans, no matter where we live and no matter the color of our skin, on the one hand, and yet on the other hand, when you were tracing the development from apes down to our line, you had apes in quotation marks. And Sibley and Alquist didn't just compare humans and chimps, but they compared humans and gorillas and chimps. And of all the relationships, the humans and chimps were the closest. Right. Closer than gorillas and chimps or any other great apes, which puts us squarely in the great ape group. So I'm curious why you have the quotation marks.
I didn't want to imply that the hominin lineage itself was still an ape. We come from an ape stock.
Well, but we still have 98+% agreement in the DNA.
There are many ways you can. The taxonomic debate about how you classify humans now that we know that we're so close to chimps is not resolved. There are some authors who would say that chimps should be Homo troglodytes or we should be pansapiens. And this was suggested by Jared Diamond in his book called The Third Chimpanzee.
That was my second question.
Yeah. So I don't mean to be able to resolve the taxonomic debate. I think the problem is when you start looking at these different regions of the genome, and each of them is telling us something slightly different. I mean, on average, yes, we're 98.8% identical to chimp, so we should take that for what it's worth, and the taxonomy will eventually be sorted out, but it's not completely sorted out right now.
When you said that we're almost in the same genus as
Again, a taxonomic problem that, is complicated.
I have a basic question about is the DNA that you're tracing coming from modern people?
That's a very good question. Is the DNA that we're tracing all from modern people? all of the work that I reported to you tonight comes from contemporary sampling, except the Neanderthal bone mitochondrial DNA. So we, there's very little hope for us to be able to recover autosomal, X chromosomal, Y chromosomal DNA from specimens that are 20 or 30,000 years old. So we're really probably going to have to live with looking at contemporary variation and tracing it back rather than looking directly at the past. For that, we've got paleontologists and archaeologists who look directly at the past. Their challenge is to know who left descendants and who didn't.
Okay. Well, then going back in time to the common ancestor and looking forward, what happened to all of the other DNA that was in other people at that point in time?
Well, that's an excellent question as well. What happened to the DNA lineages of all of the other ancestors? Well, you can think of it like this. If we're tracking a surname, for example, and there are 10 different people with 10 different surnames, and they have children, and they have just 2 children each, they have a certain probability of not having a son. And in those families that don't have a son, 1/4 of the time they won't have a son if they're having 2 children, they will not have their surname passed on to the next generation. And so that surname goes extinct. So it's a process of extinction through a random, who manages to get their offspring in the next generation. And for unilineal markers like the Y in the mitochondria, it's a pretty simple story of lineage extinction due to lack of sons or lack of daughters. The same thing can be applied to the others. It just takes a lot longer when the lineages are jumping between different ancestors in the past.
Thank you.
Over here.
Oh, hi.
From your long look at the journey up to now, which, by the way, I thought was very well summarized, do you have any speculative thoughts about the future evolution of humans generally, in brief, I understand, and specifically I'm interested in whether you see any indication of, I don't know if you'd call it subspeciation, but of the possibility of modern humans separating into 2 or more different species?
Well, that's a very tempting question to answer. I sometimes look around and wonder if we aren't already speciated. One question is, we really only, we say we should learn from our past so we don't make the same mistakes again. All we can do is try and understand the past, and I don't know how predictive that is of the future. There are a couple of things that come to mind. One is that humans are still evolving. There is some work that's just been reported showing that the effects of natural selection on the genome are ongoing. So you might hear that because of medicine and other things that we've reduced the selective pressure enough on humans that we're no longer evolving, but we are. And there are enough changes that have gone on in the last 10,000 years that show that we are still evolving. I guess the other thing to think about would be how humans have become somewhat decoupled from our environment because of our technology. As trying to trace the origin, the ancientness, or how recent our specialness evolved. And I think one of the things that makes us special is our technology. And for probably a fairly long period of time, going back a few hundred years anyway, short time in evolutionary sense, but we've developed technology that essentially decouples our evolution from our environment. However, I think with next year's lecture series on climate change, we're going to find out that we've probably created a new environment that we may not be able to decouple ourselves from. So I think that's probably something to think more about.
How about a round of applause to Mike for an extraordinary lecture? Before you all leave, just quickly, that next Tuesday, which will be the final lecture of the lecture series, which will be an evolution of disease, the HIV example, that lecture will be at Centennial Hall. and all the lecturers from the series will be there at the end for a small panel discussion of the whole series. Thank you for coming. Great job. That was fantastic.
Thanks. Oh, good. Excellent. Thanks.